Welcome to our evenings at the Homestead. We're continuing with our Seasons of Change. This is our first event for the calendar year, so we're really happy to have you here. Obviously, as you've noticed, we've gone to a virtual event here, and so that means that everybody gets to watch from the comfort of their homes. And the theme for tonight is vital signs, and I'll introduce that to you in just a few slides here. But just to show you that we've already kicked off our season with a couple really fantastic events. We started with an event describing the partnership that SCCF has formed with the U.S. Center for Coastal Solutions and talking about what that collaboration will do for us and for our communities here. We heard from our recently outgoing CEO, Ryan Orgera, talking about what he'll be doing next. And we got the opportunity to see an overview of what was accomplished collectively while he was here. Also welcomed our new CEO, James Evans, who's in the audience with us tonight. And a reminder that you probably noticed, but we set this up as a two-part event, but the way that we set this up is that you can go to one, you can go to the other, you can go to both, and we'd love to have you join us for both. But we set this up so that you can enjoy the content regardless. And a couple Zoom housekeeping notes for us all now that we've shifted to this webinar format. So what's going to happen is that I'm going to sort of frame our evening. I'm going to introduce our speakers, and then we'll have a collective question and answer session at the end. And so what you want to do is you should be able to see a Q&A button on Zoom, and that's where you can pose your questions. You can type them out to us at the end, and I'll go ahead and read them out for our speakers, and I'll help facilitate having them answer your questions. So just a reminder too, that we won't be able to see or hear our participants. And also that the seminar will be recorded and we will post this for you to rewatch again later. Again, I just want to kind of frame our evening for us. You could probably gather this from our event description, but for these two evenings, we're going to get a bit metaphorical. So we're going to look at how SCCF as an organization works towards keeping our connected natural estuarine and coastal systems healthy, much in the way that we can think about the networks of folks and technology that are part of keeping human patients healthy. So we're really using that human health lens to look at SCCF's work. And pardon just a second. So we can think about in the human medical system where you have doctors, you have nurses, you have technicians, you have any number of specialists who work collaboratively to understand what's going on in the various systems of somebody's bodies. And really what the end game there is that you use that to come up with a medical plan that either focuses on preventative medicine or actively treats or manages underlying conditions. And all the people that are tasked with this are also leveraging technology and testing to characterize what's happening for their patients. And also another way to think about this is a really important medical approach, or the best medical approaches are also thinking about not treating just the symptoms, but also treating the underlying root causes that's causing those problems. And so I'm going to give you a human medical system example. We can think about something like headaches. We know that headaches are a really common symptom that are caused by a lot of potentially different kinds of stressors, different things that could be causing them. And in order to effectively treat them, especially if somebody has chronic headaches, you really want to understand what the underlying cause is for those headaches. And I'll give you an environmental corollary, a way that we can think about that in an environmental sense. So say we have an area where we have salt marsh die-off for some reason. If you really want to effectively treat salt marsh die-off and that impact to that habitat, you likely need to understand something about what's causing that. And so you want to kind of dig into those underlying potential problems and ask yourself, is this something that's caused by maybe an invasive species of some kind that's interrupting our food web dynamics? Is there a pollutant at play? Have we somehow shifted the physical characteristics of that system? Is salinity different? Is temperature different in such a way that's outside the bounds of what the vegetation and the other wildlife are adapted to? So again, that idea of really wanting to understand the root causes. And we can think about SCCF in this sort of way, in that we've got this network of staff here at SCCF, and what we're doing, instead of focusing on the human body, is we're looking at these really complex, interconnected natural systems and trying to support their healthy functioning. And we can think about the fact that we essentially, as an organization, we've got this wealth of what I would sort of characterize as our in-house expertise, but we're also leveraging this really powerful and wealth of partnerships that we also have, and collaborating and using that to really effectively characterize what's going on with these natural systems. And so, whether we're talking about understanding the health of our wildlife, and you can see some examples, our different kinds of turtles and our birds, or if we're looking at particular habitats or a suite of habitats. And really what we're doing is we're using all that information to make our medical plan, if you will, that positions us to take preventative actions or to intervene or address current concerns. And that also means that this positions us to advocate for and work towards addressing the source of issues and not just the symptoms. And so tonight, what we chose to focus on was some examples of how, as an organization, SCCF is using science and technology to take the essential vital signs of our local and regional environment. And so tonight you're going to hear from our research and policy associate, Leah Reidenbach. And you're also going to hear from Eric Milbrandt, the director of our Marine Lab here, as I describe some of our strategies for taking the pulse of our natural systems. And so with that, I'm going to turn this evening over to Leah.
Hi, everyone. Thanks for coming to the talk tonight. I'll be talking to you about monitoring and recording the health of the Caloosahatchee estuary using our new drone, and also the past year in water quality. But first, I'm going to be going over the history of the Caloosahatchee watershed development, and I'll also be talking a little bit about ecological indicators that help us measure the health of the estuary. So in its natural healthy state, let me get my pointer here. The Kissimmee River watershed discharged into Lake Okeechobee, which passes water through to the Everglades, and discharges from Lake Okeechobee to the Caloosahatchee were relatively small. The slow-moving, wide and shallow path of the flows from the lake all the way down to Florida Bay is described as sheet flow, and it supported a variety of ecosystems, including the impenetrable sawgrass prairie plains that existed south of the lake, rich in slough landscapes, and cypress and swamp forests. With such a great variety of habitats, the Everglades provided a sanctuary for numerous species of plants and animals, and was the most significant breeding ground for wading birds in North America. The lake was around 20 feet deep, with 2 to 3 feet in variation, as the lake was able to naturally expand and contract with a wide outflow area. And Lake Okeechobee was not connected to the Gulf of Mexico or the Atlantic Ocean naturally. During the first half of the 19th century, explorers described Florida's wilderness as an inaccessible, worthless, mosquito-infested swamp that was unpredictable to settle. Draining the swamps and creating dry land that could then be farmed and built could transform southern Florida into a pioneer's paradise. The drainage began in the 1880s, starting with dredging along the Caloosahatchee River, which focused on expanding the headwaters and constructing canals from Lake Flirt through Lake Hippachee to the 3-mile canal, which connected to Lake Okeechobee. These projects were funded by state and federal funds, and the successful drainage of efforts forever changed the hydrology, soil, and vegetation of the Everglades. The drainage exposed millions of acres of previously flooded land for development and rich fertile soil, which could now be used for agricultural purposes. But the unintended consequences were flooding in areas surrounding the lake. There were devastating hurricanes in 1926 and 1928 that caused the lake to overflow its southern banks, and this caused a devastating loss of life and millions of dollars in damage. The state called upon the U.S. Army Corps of Engineers to widen and deepen the St. Lucie and Caloosahatchee waterways in order to prevent future flooding, and the construction of the Herbert Hoover Dike would cut off the natural flow of water south to the Everglades. After the hurricanes, the Army Corps focused on flood control rather than drainage, and many engineering projects have shaped the way the greater Everglades system is today. In 1935, dredging began again to widen the Caloosahatchee, and construction began on the locks and dams to provide an outlet for releasing water from Lake Okeechobee, since the Herbert Hoover Dike blocked the flow of water south. Ultimately, the Caloosahatchee River went from a narrow, winding, and slow-moving river with oxbows to a straightened, deep, and wide canal that moved water rapidly. And after all was said and done, over half of the original ecosystem's Everglades remained. Currently, the system gets stressed during times of drought and during flooding. When the lake is too high in the wet season, water is released. The Caloosahatchee can be inundated with nutrient-rich water from the lake, and during the dry season, if the lake is too low, the Caloosahatchee doesn't receive enough water. The conditions can almost never be ideal for all of the parts of the system, because the needs of the various parts of the system have different, sometimes conflicting needs, and this process can be highly politicized. Decision factors in regulating Lake Okeechobee include flood prevention and safety, urban and agricultural water supply, navigation, recreation, and the environment. Lake Okeechobee is the beating heart of the Everglades, and the rivers and canals are like the arteries and veins, moving water, which is the lifeblood of our ecosystems, around to the different parts of the system, which are the vital organs. These processes are controlled by a brain, and this brain is the various operation schedules that the U.S. Army Corps of Engineers uses to regulate flows throughout a highly controlled system of canals, locks, pumps, and levees. Currently, we're under a regulation schedule called LORS 08, which can inundate the Caloosahatchee with high flows, which led to the harmful algal blooms we experienced in 2018. A new regulation schedule called LOSM, the Lake Okeechobee System Operating Manual, should reduce stressful high releases to the estuary. Once other projects are completed, such as the water storage south of the lake, the EAA reservoir, and along the Caloosahatchee, the C43 reservoir. The LOSM is currently under development, and I think we'll be hearing more about that at our next evenings at the Homestead event in February. So too much flow or too little flow to the estuary can disrupt the ecosystem and cause many harmful effects. These can be thought of as diseases to our estuary, and the symptoms can have negative effects on the environment and wildlife, the economy, and human health. We use 3 indicator species, seagrass, tapegrass, and oysters, as a way to guide how much flow is needed for a healthy, functioning ecosystem. These guys are like biomarkers such as cholesterol or white blood cell counts that need to be within a certain range. Like I said, water flow is the lifeblood of our ecosystem, and it needs to be in a specific range to provide optimum salinities for our indicator species. These species are important because they provide habitat and ecosystem services, and they're sensitive to environmental fluctuations. When there's too little flow, salinity is too high, which causes stress to the oysters and is stressful or lethal to tapegrass in the upper estuary. When there's too much flow, decreased salinity in lower estuary causes stress to seagrass and oysters. So one of the things that we do at SCCF to kind of monitor the disease progression and make sure that we're advocating for a healthy ecosystem is the Caloosahatchee Conditions Report. So this is a weekly report that comes out on Wednesdays, and we meet every week with the City of Sanibel, Jane Ding Darling National Wildlife, the City of Cape Coral, Lee County, and others to produce a technical scientific report. And we use a variety of data sources to and kind of synthesize them and put them all together into one source. And we also make a recommendation to the U.S. Army Corps of Engineers on behalf of the Caloosahatchee estuary. The report includes things like flows, salinity, cyanobacteria in the river, water clarity, red tide, fish kills, and impacts to wildlife, especially birds, which are sensitive to red tide. And we also include an aerial image each week because it's easier to show people what water clarity and quality look like than it is just to tell them some numbers. Another report that we put out each week, we put it out on Friday. This is for residents and for visitors to the island. It's less technical and more educational, and it kind of guides people to say, are the beaches safe to go to today? Is there a lot of red tide, or is there anything going on water quality wise. So it's quick and easy to get updates for conditions around the island, and there's some educational articles in it periodically. It features an explanation of the Lake O releases and watershed runoff, because you can't really talk about the Caloosahatchee estuary without talking about Lake Okeechobee. The current aerial image off of Sanibel's Lighthouse Beach Park, and updates on algae blooms and any current events related to water quality. So this year we received a grant from the Coastal and Heartland National Estuary Partnership to buy a drone, and ever since we received that drone, we've been flying at Lighthouse Beach Park every week to capture aerial images. We've been putting these images in the Caloosahatchee condition reports. We created a 360-degree virtual tour website. It's also part of the weekly conditions update. And over the course of the past 8 or 9 months, we've documented a few water quality events, such as cyanobacteria blooms at the Franklin Lock and dams, and drift algae and macroalgae accumulation on the beaches. And we also got some pictures of the effects of Tropical Storm Elsa. This also gave us the opportunity to collaborate with the city of Sanibel on some of their water quality projects. So here is just an example of the 360-degree website, virtual tour website. So when you click it, you zoom into the latest image, and you can see on the top here that it gives you some information about when the image was taken, and also what the tide was like at that time, and also the 14-day average flow. So then you can just kind of scroll through the images to see what water quality has looked like recently and how it's changed over the past few weeks. You can also go all the way back in time. So if you want to see what it looks like, you know, at the beginning of the wet season, before we got a lot of rain, you can see that the water looked really beautiful, and the 14-day average flows were within the optimal range, which I'll be explaining what the optimal range is later. You can also look around the entire island to see, you know, what water looks like. When 14-day average flow gets a level higher, like this one is 5,355, that one's in the damaging flow envelope, and you can already see that it causes the water clarity to be reduced. So this next part of the talk, I'm basically going to be going over the whole year 2021, what water quality looked like. But first, I kind of want to orient you to this graph, which is going to be pretty much on every slide. So these are flows in CFS coming out of either S79 or S77. And this is the entire Caloosahatchee watershed. It encompasses a drainage area of over 1 million acres. So S77 is the structure that controls flow from the lake into the freshwater part of the Caloosahatchee River. S79 marks the beginning of the Caloosahatchee estuary. So all the flows from here to here, including Lake Okeechobee, all eventually end up in S79. So on the graph, the flows from S79 are the ones in black, and the ones in this pink purple color are S77. So these are the flows from the lake. The blue line is the 14-day average flow, which is kind of like the ecologically relevant time span for flows. So if you get 1 day of really high or really low flows, that's not going to have a huge effect. But if you have 2 weeks of really high or really low flows, that's when you're going to start to see some negative effects. And then this red line is the Lake Okeechobee stage elevation, just for reference. And the green box here, that's the optimum flow envelope, which is between 750 and 2100 CFS. So right now, all that you need to know about that flow envelope is that's the flow envelope that we want to be in most of or all of the time. So starting the year off in January 2021, it's not shown on the graph, but before the year started, we were actually 16 weeks above the optimal flow. So we were more up in this range. So we started the year off kind of not great, but then it got back down into the green part. And we also started off with some red tide, which actually ended up lasting the whole year, basically. In February, lake levels were kind of too high for the beginning of the year, which prompted concern about the start of the wet season. So if the lake is too high at the beginning of the wet season, we get worried that they're going to start doing major releases to the estuary. And water couldn't move south because the water storage areas south of the lake were too full, and so the Corps was struggling to reduce lake levels. We were in the optimal flow envelope at this time, so that was good, and red tide continued to have effects on wildlife. There was also massive die-offs related to cold fronts and high winds, which is exactly what happened this week. So if you go on the beach this weekend and you see a lot of, I went today and I saw a lot of dead scallops and sea urchins, that's related to cold fronts and high winds. But there was also, if you can recall, if you were here last year in February, there was a lot of royal terns dying on the causeway, and a lot of them were tested for brevetoxin, which is the toxin in red tide, and they came back positive for it. They also came back positive for bacteria. And on top of all that, there was really high winds, so they were just getting they were getting into trouble on the causeway and getting hit by cars and dying, and that was due to 3 different environmental factors. We also saw a lot of fish kills due to red tide. In March, we started seeing some algae on the river. This is S79, and some macroalgae near Shell Point. We were kind of plateauing near the top of the optimum flow envelope, and red tide was ongoing. And I can't go without saying that on March 26th, there was the Piney Point environmental disaster, which was a phosphogypsum wastewater reservoir, which began leaking. And so in order to prevent that from becoming worse, they started releasing contaminated wastewater full of nitrogen and phosphorus into Tampa Bay, which exacerbated the ongoing red tide in that area and resulted in the cleanup of over 1,600 metric tons of dead fish by the end of the bloom in July. So we were fortunately spared from devastating massive blooms such as this year. In April, there was a macroalgal bloom, nearly 100% coverage of Caulerpa and jay and ding darling. So Caulerpa is a type of macroalgae, and it basically can smother seagrass and block the light. And we also saw some cyanobacteria on the river. We were still in the optimum flow envelope, and the lake was receding. So May, spring kind of comes along, and we get a lot of reports of algal blooms. So we saw it at the Davis Boat Ramp, which is on the river. This is S77, and we also saw it at S79. And we saw some drift algae accumulation at Bunch Beach, and there was a Trichodesmium bloom. Trichodesmium is a nitrogen-fixing microalgae can be really gross when it's blooming. So this was in some canals on Sanibel. But the water clarity at Lighthouse Beach Park was still really beautiful. So spring blooms like this, spring blooms of macroalgae, can be caused by increasing temperature, increasing sunlight, and obviously nutrient availability. And they can be somewhat ephemeral in nature. This year, these lasted for several months. But it's that combination of light, temperature, and nutrients that drives these blooms. So in June, we finally got the lake down to a level where we weren't so concerned about major releases during the wet season. So that was a really good thing. We were happy about that. We were still seeing a lot of issues with Trichodesmium and macroalgae blooms. So some of the blooms that had occurred the month before started decomposing in Matlacha, which caused anoxia and hypoxia. And we were still seeing strandings on the beaches, but water quality still looked really pretty. And you can see that we're still in that optimum flow envelopes, and the water is mostly coming from the lake during this time. That's a pink line. So July, summer, wet season is going full blast, and you can see that now we're getting outside of that 14-day average flow, or the 14-day optimum flow envelope. And since wet season had really begun, they stopped releasing water from Lake Okeechobee. So all of this water is coming from the watershed. We didn't get as many reports of algal blooms, and Corinia was lingering, but it really remained concentrated up in the Tampa region. Tropical storm Elsa hit on July 7th, which was a major rain event, and it caused water to get released from the Sanibel River and out into Pine Island Sound. So you see the super brown tannic water from the Sanibel River being released out here into this really bright blue turbid water from the hurricane. So it's quite a striking image, very temporary. In August, we continued to see algal blooms and fish kills from red tide. Tropical Storm Fred and Hurricane Ida were in the Gulf of Mexico, which were major rain events and wind events, but overall we were really lucky not to get any direct impacts from tropical storms or hurricane activities this year. And in August, there was no more cyanobacteria on the river. So September, we start seeing a big jump in rain and flows from the watershed. You can see that this week, the 16th through the 23rd, in this area here, there's this is the average rainfall in this Caloosahatchee watershed. It was like 2 to 5 inches in some areas. So on the 17th, the water is looking pretty clear, and then by the end of the week, the water starts to look really brown, and that's that damaging flow envelope that we're seeing here. So these changes can really happen in a matter of days when you get these really big rain events. So the rest of the year, I'm just gonna put that out there. Not really a lot happened. It was fairly quiet. The dry season began, so we started seeing flows again from the lake, but it was all kept within the optimal flow envelope for the most part. And we started, these lake flows, I think, prompted some cyanobacteria on the river again, but nothing major. And water quality remained poor for the rest of the year. And we continued to see a patchy, lingering red tide in Southwest Florida. So overall, there were no major algal blooms, but red tide was present on the entire coast, or on the coast for the entire year, with impacts on wildlife like fish kills, intoxicosis, and birds and sea turtles. We were concerned about high lake levels at the beginning of the year, but avoided major lake releases during the wet season. And water clarity declined during the wet season due to local watershed runoff. 61% of the flows to S79 came from the watershed. And in May, June, July, and August were the months with the most reports of water quality issues like macroalgal strandings, die-offs, and decomposing algae. So this is a graph of flows from S79 for the past 5 years. The green areas highlighted are, I highlighted the wet season from June to November. And then the pie chart on each section shows the number of days within each part of these flow envelopes. So there's damaging on the low, stress on the low side, which we don't see that often in the wet season. Optimum is green, stress is yellow, and damaging is high. And basically the main thing that I want to point out on this graph is that this year we spent the most time in the optimum flow envelope during the wet season than we have in the past 5 years. And you can see in 2017 and 2018, we spent a lot of time in the damaging high flow envelopes, which resulted in some of the worst algal blooms that we've seen in recent years. So now I'm gonna kind of explain some of the things that maybe you had questions about, indicator species and what are the flow envelopes all about. So we have 3 indicator species here: oysters, seagrass, and submerged aquatic vegetation, tape grass. So they're immobile, they're sensitive to environmental change, and they're relatively easy to measure. So tape grass and seagrass stabilize the sediment. They improve water quality and provide habitat for many organisms and act as a nursery for many juvenile species of recreational and commercial fishes. They're also an important part of the food web. Oysters filter and improve water quality and provide important habitat in the estuary. So these are ecologically driven flow envelopes that I've shown you in previous slides. They're based on the preferred salinity of these 3 species. So this is a chart showing that tape grass likes to be in salinity less than 10. The oyster likes 10 to 25, and shoalgrass or haliduli likes 15 to 45. So when the 14-day average flow is in the optimum range, the suitable habitat for each species is maximized. And when the flow is outside, like if it's in the stress and damaging flow envelopes, that range is restricted and negative effects can occur when the species, which is immobile, is outside of its preferred salinity ranges. And the recover flow targets are based solely on salinity ranges, but other environmental factors can affect the growth of seagrass as well. So seagrass require light for growth and survival, and low water clarity can reduce the amount of light reaching the seagrass, a process called light attenuation. Low water clarity can be caused by a combination of turbidity from wind and wave action, chlorophyll from phytoplankton, and color from organic substances like tannins. So these three factors are influenced by tide, weather, flows from the watershed, and flows from Lake Okeechobee. So nutrient loading from high flows from the lake and the watershed are rich in nutrients, which can promote the growth of epiphytes, which are organisms that grow directly on the surface of seagrass, macroalgae, when you get more nutrients, and then finally phytoplankton, which can block out and compete for light with seagrass. These effects can happen relatively quickly. So this is November 22nd, when the 14-day average flow was in the damaging flow envelope, it was in the stress flow envelope, 2,242 CFS. And then just a couple of weeks later, after the 14-day average flow went back down into the optimum flow range, you can see that water clarity improved quite a lot. So now to get back to our analogy on human health, the Greater Everglades system is a highly altered and managed system with issues with the quality, quantity, timing, and distribution of water across various stakeholders. And we collect data about the symptoms of the ecosystem each week and distribute it to various agencies and the public. And the recommendation that we make on behalf of the estuary for flows are like a prescription that can be used to maintain correct salinities in the estuary. And I didn't get time, or I couldn't talk about this today, but the completion of the Everglades restoration, or SERP, is going to be the cure once all the projects come online. It's not going to be a perfect system, but it should be a lot better. And this QR code, if you want to sign up for our newsletters that I talked about today, you can scan that now.
So again, so thanks to our first speaker, Leah. It was great to hear her talk about water flow as a vital sign, the kinds of impacts we think about from too much or too little of that. We got that great overview of essentially a water quality panel over, spanning the whole year. A couple really amazing tools, those weekly condition reports and the drone imagery. So thanks again to Leah. And again, a reminder that now we're moving on to our next speaker, which is the director of our Marine Lab, Eric Milbrandt. And again, as Leah had noted, we're keeping an eye on questions, but we'll do a combined question and answer session at the end. So take it away, Eric.
All right. Hey, thanks, Carrie, for the great introduction, and Leah for sort of laying a lot of the groundwork for my talk. So ecosystem vital signs, we're making this analogy because there are many parallels with human health, and we're keeping a finger on the pulse. And so my talk is largely focusing on different tools that we're using. I think the drone and the imagery has become a powerful aesthetic tool, but we started really collecting a lot more data in the water and in the air in 2007 when we launched an observing network. So I'll describe a little bit about that to you today. We're in the process of renewing our observing network through new technology and better technology and an increased number of parameters that will help us better both describe and explain the current condition of the estuary and eventually get to the point where we can model future conditions. There we go. I was pressing the down arrow instead of the right arrow button. So water quality, of course, is a big concern for Floridians, probably one of the reasons why a lot of you are tuning in tonight. We've had a pretty rough last half a decade here. These images are from various parts of the state of Florida and really are happening here as well. Algae blooms are the disease. They're difficult to treat as the disease. You know, you can try and figure out the causes, which is probably much better. There is a group of engineers and scientists that are working on trying to clean up these types of situations. But the reality is the scale is way too big, and you're going to have many secondary effects if you try and treat the bloom. So what we need to do is really focus on the causes. So one hot piece of history that I will share with you is that the treatment of an algae bloom did happen in a large scale in Florida. In the Gulf of Mexico, they decided to put copper sulfate in the ocean, and you can imagine it took care of the bloom, but it also killed everything else, just like what would happen in your pond behind your house. So if you do have, this is just a public service announcement, if you have a lake management company, tell them not to use copper sulfide. It's not good, and it's widely used and legal in ponds and so on. But I want to just describe to you what the different types of blooms are, because we're focusing a lot of our research efforts on collecting and monitoring algae, and they're microscopic until they become a big problem. Okay, so we're talking about normal concentrations of cells, 1,000 per liter, become millions, tens of millions per liter. In this case, in the freshwater parts of the Caloosahatchee, you have cyanobacteria, also called blue-green algae, and Microcystis aeruginosa is the colony-forming cyanobacteria that's caused by excess nutrients, and many attribute the bloom of 2018 to the discharge of highly nutrient-rich water from Lake Okeechobee, along with the bloom, essentially seeding the freshwater Caloosahatchee and causing a massive problem. Switching gears a little bit to the Gulf of Mexico, we have Florida red tide, and this is a dinoflagellate that produces a toxin that kills fish, hence the reports of dead fish that are happening when we do have the blooms. This great photo was taken on a parachute with a person with a propeller on their back, and he works for National Geographic and captured this great pictures. So red tide is a little more complicated. You have to imagine these are not jars with algae growing in them. These are open systems, so they're complicated, lots of processes at work, tidal forcing, there's ocean currents at large scales, including the Florida loop current, that cause the blooms to initiate. There's much of our evidence about red tide. The blooms initiate offshore, they're transported inshore, and more evidence now is surfacing that the cells use the coastal runoff as their fuel. So the other sort of problem with algae blooms is you get side effects. Okay, so like if you are overweight, maybe morbidly obese, you're going to have heart problems. You're gonna have cholesterol, high cholesterol. You're going to have other side effects that are associated with that condition. So similarly, the side effects of too too much algae on the beach, you can't go to the beach because you have to cross a mound of rotting algae. With red tide, I described before, they have a powerful neurotoxin that affects fish in a disproportionate way, but it also can be aerosolized in the surf zone and affect people. But at least people can move away from it and go inside, which isn't, a great solution, but at least you're being affected, and the fish, unfortunately, can't move away. And then the other thing that we've talked about a little bit is the algae blooms can cause very large dissolved oxygen minimum zones. So areas where there's just no oxygen. Anything that lives in the water needs oxygen, just like we need oxygen. So when you remove it, because you have so much algae decomposing, bacterial activity, so on, it causes extremely large hypoxic zones. So what are some of our tools and tests that we use? Similar to a EKG or a lipid panel, we need to figure out what's going on in the ecosystem. And to do that, you need constant monitoring of water quality conditions. I think Leah did a great job of outlining our flow, how we need to get the flows right. And that's really an engineering problem. But also when we get the flows right, we get conditions that are favorable for everything. And that includes the seagrass, the oysters, and it can prevent algae blooms. Whereas you get the wrong flows, you get conditions that are favorable for their growth. So the important part of this is that our water quality measurements are now more and more being automated, similar to the way that you have a tracking watch, a Fitbit measuring your steps or your heart rate. These things are not something you have to wake up in the morning and turn on and turn off at the end of the day. So they're providing data all the time from multiple sites, and what we call this is whole system intelligence. So this is these sites on the map. That's our logo, of course, the shorebird standing on water within a marsh, which is very symbolic for us. But those sites are places where we measure water conditions every hour. And in this graph, it's just water temperature. So fairly straightforward. You have a peak in the middle of the day where it's the warmest, and then at night it cools down. And so you can see that pattern replicating at all the sites. However, some of the sites upstream, for example, it gets much warmer, especially in the summer. So those are things that we pay attention to, and it's like an EKG. So we can look at these graphs and help figure out what's going on in the system. So these are the different water quality parameters. I borrowed this slide from my colleague AJ. Salinity is the amount of salt in the water. Water temperature is self-explanatory. Dissolved oxygen, we talked about that. It's the amount of oxygen in the water. Depth of the sensors isn't really that useful until you start thinking about transport of algae blooms. Is it a high tide or low tide? And where are things going? Chlorophyll, that is the amount of microscopic plankton in the water. Turbidity is the transparency of the water. It's usually affected by silt, mud that's been either resuspended by wind is the most common cause here, but also at times Lake Okeechobee can be a big source of that silty water. CDOM is the colored dissolved organic matter. So that's just like tea. If you had a tea bag and you put it in hot water, you would get that tannin coming out of it. It's the same thing in our estuary. And then at two sites, I talked about nutrients a little bit, but I'm going to talk a lot more about them in the next few slides. We are measuring nitrogen and phosphorus recently because of an initiative to pilot test new sensor technologies. And we are one of 5 labs selected in the Gulf to do this pilot. So here's a neat trick that we programmed in with a web designer and a data guru. Our website has the ability, it has a warning system when certain thresholds are exceeded. So when Leah was talking about tape grass, it's a freshwater under submerged plant, provides lots of habitat for fish and blue crabs and so on. If the salinity gets above 10, it does not last more than a few days to a couple weeks. So we have a warning system on our website that tells you when conditions are being exceeded. And the main driver of that is, as Leah said, was flows. But yeah, let's talk about causes. Just, preventative medicine is the way to go. And we're not going to treat these algae blooms. It's way too expensive. They're way too big. So what causes them? Most of the science tells us that nitrogen and phosphorus, too much nitrogen and phosphorus will cause phytoplankton to grow with no limit. And those blooms can be held in check at times in certain places, like they fuel much of the global ocean, but it's not an overgrowth. And a lot of times there's a balance between things that are grazing or eating the phytoplankton in the ocean or filtering out the phytoplankton. So, I don't want to disparage phytoplankton everywhere, because it's a really important part of the way the world works in our oceans. But unfortunately, when you have we produce nitrogen in massive quantities to fertilize crops, and those crops are fed to cows, and the cows are on the landscape, and that water runs off into our ocean. So it's something that we need to address, and there's many ways that you can do it, including building more wetlands and fixing the Everglades, as Leah's slide pointed out. So many of the drivers of the blooms are actually also linked to climate change. So changing snow and rain patterns. We don't have snow really here, but stronger storms that last longer, the damaged corals in the Florida Keys and elsewhere around the world are bleaching because of rising sea level, rising sea temperatures, warmer oceans. Those all fuel harmful algae blooms. And then, of course, we have warmer temperatures on land as well. So one thing we know, and this is data that we collected, I've smoothed it so that it's hourly data is very, it looks like an EKG, right? It goes up and down. But this has been smooth. So this is like a 15-day average from hourly samples collected in 2015, and the pink line is nitrate. So that's an inorganic form of nitrogen, and it's usually associated with fertilizers and also runoff. And we can see that the algae, the green line, closely follows the nutrient concentrations, and actually it's not just concentrations, these are what we call loads. So it's the amount of barrels of nitrogen, not necessarily just the concentration in the water. So those are that's really strong evidence, and so any way we can try and address that nitrate by letting water sit on the land longer, including some of the swales beside your house, in front of your house, really important stormwater rules that were created to help absorb nitrogen and prevent blooms. So the other analogy that I think is interesting, this is a, if you look from left to right, you're looking at the coast. First of all, there's Sanibel and our red stars where our instrumentation is. So that's a model that is showing where water is going. And in the middle is the watershed, and Leah showed maps of this. So this is the area that drains into the Caloosahatchee. And then on the right is the lake. And so what we're currently doing with our colleagues is we are merging all these three models to provide a complete whole system modeling approach. And the analogy of this to me, because I have allergies, I didn't know until I was probably in my early 30s what would cause me to get hives or make my nose run. And so I went in and I got tested and they scratched me with all these different allergens and figured out that I was allergic to dust mites and I was allergic to mold and all of these things. So what this model will be able to do to me is help us figure out what causes the blooms by identifying where the blooms are starting, where they're being transported, and where they're ending, and looking at the conditions during those events. So our observing network started in 2007, and it's about the same time that the first iPhone came out. So we are now, this year, thanks to the city of Sanibel, Lee County, Paul and Lucy Roth, and Deborah Lagoris, and another anonymous donor, we're able to take all of our equipment, that was basically our iPhone, first iPhone, and we're going to put it on the shelf, recycle it, and we're going to replace it with the 13. Okay, so this is all brand new stuff with new modems, new sensors, better technology, better data. So as many of you have probably heard, there's no more 3G. So that's a problem because all of our data for that whole time period was being sent through little email packets over 3G technology. So now we're 5G. So we've upgraded there. The sensors haven't really changed a lot in terms of the basics. The salinity is a measurement that's been around a long time, but dissolved oxygen and just the way that that the technology works, it's simpler and it's smaller usually. And this is one good example of that. We have our old sensors on the left are kind of like a size of an oatmeal box or can. And so we had one measurement that would measure the amount of algae in the water. Well, that same oatmeal can, if you will, now can measure 4 different parameters. So we're going to be able to look at not just the total phytoplankton in the water, but we're going to be able to look at the blue-green algae specifically, and turbidity and color dissolved organic matter. So that's the end of my talk, but I do want to thank you all for being here. And I know we're all probably in it for the birds. And this is a shot from Hemp Key, where I've been working with some colleagues to try and restore habitat there. And we're using a whole system approach to protect and care for Southwest Florida ecosystems. And at this point, I'm going to hand it back off to Carrie, and we will proceed with Q&A.
Yeah, well, hello, everybody again. So thanks again to Eric. I really appreciate how he continued our conversations around vital signs and how they tell us about water quality, how that might contribute to algal blooms, how we can think of those algal blooms as a symptom, and why we need to really treat the underlying causes and not just the symptoms. We got that great insight into the Recon network as the technology that we can use to characterize all that, but also the fact that we're moving into more cutting edge territory with the Recon network, which is really fabulous. So before I move into Q&A, I was reminded that in the midst of all the multitasking, I didn't get an opportunity to introduce myself. And unfortunately, again, you can't see me and I apologize for that, but the voice that you're hearing belongs to me. My name is Carrie Schuman. I am the Coastal Resilience Manager at SCCF, and I'm really happy to be part of this evening. So we'll go into Q&A. We have a couple questions. Oh, I see at least one more question coming in. And so what I'll do is I'll go ahead and read these. And just so you know, at the end, I'll also tell you a little bit about our next related evening. So, you know, if you want to stick around to hear about that, I'll be happy to tell you. So our first question is from Barb. I'm not going to attempt some of these last names, so I apologize. But Barb asks, can you please address the birds that died on the causeway bridges? And I believe that's a question that's directed for Leah.
Other than what I said in the talk, you know, there was the three factors. There was brevetoxis symptoms. They had bacterial infections and also high winds. And them being on the causeway, they ended up getting hit by cars and dying from that, I think, mostly. I don't know if you know more about that topic.
Yeah, I do. We were able to write a grant proposal, and it was great. It was a great community effort because our colleagues at Crow were harvesting, and this kind of maybe a little too biological for some, but they were keeping the carcasses and they were like separating the tissues. All right, so we were able to send those off and get them analyzed for brevetoxin. At the same time, a group of volunteers and fishing guides collected fish. And so a lot of the royal terns, they eat at a specific part in the food web. Okay, they eat small white baits and other small fish. And so we were able to collect over a period of time a lot of fish. We had enough money to analyze all of those. And so the data we've got back is that, yes, there's a lot of brevetoxin in the food web. Yes, the birds had a lot of brevetoxin in them. Logical conclusion is they were very sick, and the causeway was a convenient place for them to land. Although, you know, most terns, like from my understanding and talking to my shorebird colleague, they like, you know, big, wide, flat beaches. And so it could have just been a high tide in the middle of the day and it wasn't space. But we also found that the concentrations of the toxins around the causeway islands were high, and maybe that's part of the reason why they were getting sick on the bridge.
Okay. So the next question is from Richard. Richard says, it looked like watershed runoff was responsible for some of the worst water quality events. If that's the case, how would redirecting lake water into the Everglades significantly reduce this problem? And I think I'll direct that to whomever would like to answer.
Yeah, I'll take that one. So Richard, that's a really great observation. And I've also noticed that too, but redirecting lake water towards the Everglades is going to be good for the Everglades, and it's also going to reduce the pressure on the northern estuaries to have that water released to them. But there's over 60 projects in the Comprehensive Everglades Restoration. And one of those projects is the C43 Reservoir, which is a 170,000 acre-feet reservoir on the Caloosahatchee River that is going to be able to store excess water during the wet season and release it after it's been treated back to the Caloosahatchee in the dry season. So that's one of the many solutions that we'll have to reduce the amount of watershed runoff from our watershed into the estuary. But unfortunately, that only accounts for 38 to 42% of the storage needs that we have in our estuary. So there is still a lot of work to be done as far as getting that watershed runoff stored and treated before it gets put into our estuary, and to help with the timing of flows. I think that's pretty much all I said about that.
Our next question is, what are the major points? And this is from Robert, and he asks, what are the major point source pollution areas on Sanibel and Captiva Islands?
So we've been studying water quality on the islands for a while. We've identified where the hot spots are, and we also have a system to manage water that is called the Sanibel River, Sanibel Slough. Unfortunately, that water is been listed as polluted by the state. It's an impaired waterway. So a lot of it is from legacy nutrients from when we were on septic tanks. We are now, of course, on central sewer. Most of us, there are a few, but they are converting to central sewer. Golf courses are the #1 land use type that are the biggest sources or hotspots. And that's largely because they use treated wastewater. One of the strategies to get rid of treated wastewater, you can't just dump it in the ocean, is to let it sit in ponds, and then it also gets spread on the landscape. And again, like our stormwater rules, these are strategies for us to reduce the nutrient concentrations. So it doesn't surprise me that that the golf courses are big, are hot spots. So the Sanibel Slough and the 2 exit points, mostly the point that's in the canal system on the east end, is the most is the point source, and it's the way that we manage water is to protect the wetlands. But when, especially the east end, it has some infrastructure that was built before the Sanibel plan was passed. A lot of the old developed areas are on the east end. So it's difficult to sort of go back in time on sort of land uses and creating more wetlands, because we've done a great job. We've protected a lot of wetlands. We bought a lot of property. That's kind of what we've done for the last 50 years.
Okay, so first of all, I want to thank everyone. I see that there's also some just generous and very great comments here as well. Linda asks, are you aware of any politicians who are interested in your valuable data?
Yeah, I mean, I'd say that all of our council members on Sanibel are very interested in water quality. The city of Sanibel supports our work and purchased a sensor package for us. So we've had great leadership from our current mayor, Holly Smith, and they're also engaged in discussions with other West Coast stakeholders to make the management part of our water quality that's handled by the Corps of Engineers and the Water Management District. They know that good water quality is also good business and good for everybody. So yeah.
Okay, Kim asks, specifically for Eric, when you get an alert that salinity is outside the threshold that you care about, is there anything that you're able to do about it?
Well, we could, yes, I was going to answer facetiously, but because I know Kim very well. But what we do is we tell everybody that, and these are not through scientific publications that take 3 years to get to the public's attention. We are constantly talking to the media. We talk to the other managers in the region. We produce a report where it's communicated. So those threshold exceedances like salinity especially hasn't happened in a long time, and that's because we haven't had a drought, and we have a lot more of the management team that manages water paying attention to us. They have a lot of other interests they got to pay attention to, including water supply, drinking water, agriculture, the East Coast. So it's getting a seat at that table, and that's important.
Okay, let's see. I might grab at least a couple more questions for us. So Kevin asks, any plans to add more in-situ nutrient sensors with the new Recon equipment?
Hey, Kevin, glad to see you're tuning in. So right now we are planning on adding we have equipment at Fort Myers that may remember, and then at Shell Point. And the modelers have asked us if we would put a nitrate sensor at S77, which is just below Lake Okeechobee. So yeah, it's we're expanding slowly. It's obviously, I mean, we're limited in terms of how many people we have. So we're sort of close to being maxed out, but you know, with this nutrient sensor program, you know, we're one of 5 pilot test sites. So that's really exciting and good for the organization.
And I might grab one more question for us and then tell everyone about our next event. So the next question is, what is the quality of water from runoff into the Caloosahatchee River?
What is the quality? I mean,
Yeah,
depends on the day, honestly, or what time of year. It's very dynamic. It changes. We do have a lot of septic systems in the upper urban part of the tidal estuary. Today, and I would actually say for the last several months, even I would say maybe almost the last year, since we had the last red tide event, the water's clear, it's clean, looks good. I know Leah described several events that happened during the year that, you know, are not perfect, but.
okay, well, so I didn't get to every question here, but I do just want to really thank everyone for your engagement, for your comments, for your interest, and I want to thank our speakers again. And I just want to tell you just quickly about our next evening. So for our next evening, we're going to explore some examples of taking those vital signs, like the ones that you heard about tonight, and use them to inform actions that we take and the plans that we make once we understand how our ecosystems are doing. So we, of course, you could see that with our Marine Lab folks here, that we have a lot of people here at SCCF who are instrumental in steps all along the entire process, but you're going to be hearing from 2 of us who are a bit newer and who you may not know as much about. So first will be Paul Julian, who works in partnership for both SCCF and the Conservancy of Southwest Florida as a hydrological modeler. He's going to tell you what that is, why it matters. And then you'll hear from me. And again, my name's Carrie Schuman. I'll talk about my position, which is relatively new as Coastal Resilience Manager, and how coastal resilience really fits into maintaining healthy, flourishing coastal systems into our future. And so again, that event is on February 15th. And I just really want to thank you for your engagement and for joining us for this great evening. We're very happy to have you.